From Microbe TV, this is Immune, episode number 85. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, everybody. Welcome back.
Is it fall there yet?
It is, but it's warm. The leaves are changing, which is really beautiful, but it is warm. It's kind of like late summer.
It's 25 C here, and the humidity is really low, 64%.
Yeah, it's beautiful.
Also joining us from Cleveland, Ohio, Steph Langle.
Hi there. Hey, everyone. Yes, like Cindy, it's warm, but I can see some color changes in the leaves. And so I think it's our last warm couple days before it gets colder.
I think a couple of days is the official fall, right?
Yeah.
21st?
21st,
right?
something like that. The fall, what is it called? The fall equinox or the fall solstice? Summer solstice, fall equinox.
I think fall equinox.
You see, I'm not a climate scientist.
Somebody can email us.
I'm going out of my lane.
I thought the equinox is when it's equal day and night, which happened before.
Before fall? Before official fall?
Could be right.
Anyway, we'll get lots of corrections.
Yeah.
Put them in the comments below if you'd like. But be nice. That's the key.
Yes.
Because we're not climate
Well,
scientists.
it does say Sunday, September 22nd.
Okay.
8:43 a.m. is the autumnal equinox.
Autumnal equinox. There you go.
Cool.
Or
all right,
otherwise
thank
known
you.
as fall.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It's also warm, but lovely, and moving towards fall, autumnal equinox here too.
Brianne, there seems to be a character on your windowsill. Is that a windowsill?
Oh, it is.
No, it's a blackboard, right?
Back here? No,
Yeah.
there's a windowsill. And
Who's your friend?
I have Einstein.
Oh.
And he actually thinks. He points to his head and thinks. And he's solar paneled, so that's why he has to be by the window.
So his arm moves?
Yes, his arm, he goes like this.
Oh,
that's
cool.
And taps his head if he is getting sunlight.
Nice. I love stuff in the background like that. I just added a virus here.
It's blurred because it's focused
Which-
on your face. But what virus is that?
It's actually
Polio? We don't know.
it's a terrarium. It's meant to be filled with dirt, and you plant things in it. I think it came from one of those stores.
Oh, okay.
So home goods stuff. But it's blurry because I have the thing set to blur the background, so that puts more emphasis.
I
on
also
me.
have one of those terrarium that is an icosahedron at home.
I was going to say, what type of virus is it?
Mine's an icosahedron with string lights inside.
It's exactly the same thing.
Yeah.
It's got string lights, but they're often. I just illuminated it with. I might put them all here in the shelf, but it's all going to be blurry. That's the whole key, right? The blurry effect is very popular to do because it focuses on the speaker and just puts everything else in the background, so you don't get distracted. Right. Same thing at the incubator. I blur the background. Okay, enough of that.
I'm not getting distracted.
I'm always thinking of, there's always somebody in the comments on YouTube who's like, when do they start talking about the paper? How long do I have to scroll to, go past all the chatter? But other people like the chatter. So.
Hey, you can scroll. That's the thing.
Yeah, that's the beauty of it.
You can do anything you want.
Something for everyone.
Well, you got to be careful with the other comments because anyone can come and say something and they may not even watched it.
Yeah.
They just want to be annoying.
I'm unfazed.
If in fact you do happen to like these conversations, even with the diversions, we'd love to have your support. We don't do ads on our programs, as you may have noticed. Why not? They're disruptive. We're trying to teach you something. Could you imagine going to a lecture and then the professor after 10 minutes says, I just want to pause and tell you that this lecture is sponsored by. It would be pretty annoying.
Yeah.
And so I don't want to do that on these programs. That was the original idea of Microbe TV. So we depend on your generosity, financial generosity. Although I heard recently that you can donate your airline miles to Microbe TV if you want to.
Hey, we're looking to do more recording at conferences.
That's right.
Yeah.
So that would help us pay for flights, and you still get a tax deduction. In the U.S., it would be a federal tax deduction. But you also can do other things. And if you go to microbe.tv/contribute, we have different ways that you can do that. I think everyone likes to have their miles, but maybe some people, it's a no-brainer giveaway, right? It doesn't cost you anything.
Yeah.
Okay, so today we have a snippet and a paper, and I will do the snippet. It's a very cool paper.
It's not a small paper, but
I'm going to try and
Awesome.
summarize it because I think I can, bypass the method details because I don't know anything about them. I can tell you the summary, but it's a Nature article just published in July. Immunological memory diversity in the human upper airway. And this is largely from the La Jolla Institute for Immunology and the University of California, San Diego, and also Seoul National University in Korea. The first three authors contributed equally: Sidney Ramirez, Farhood Faraji, and L. Benjamin Hills. And the senior author is Shane Crotty. This is all about what's happening immunologically in the human upper airway. And, much immunology is done by taking blood and looking for immune responses. Taking blood is relatively straightforward. But if you have a respiratory tract infection, you want to know what's going on locally. And the upper tract is where things begin typically. And it can be sampled, as you will see in this paper, but it has not been sampled very much. In particular, in this paper, they're looking at immunological memory, which is really important. That has not been studied very much up there, and specifically tissue-resident memory T cells, TRMs. They don't circulate. They stay in the tissue where they originated. And Donna Farber at Columbia has done a lot of work looking at these cells from organ donors or cadavers. If you get the cadaver quickly, you can get relatively fresh tissues and look, and she has characterized lots of T-resident memory cells that are organ-specific.
And since they don't circulate, you aren't going to find them in your study from the blood. So you have to do these studies of other types of tissues.
We also know about tissue-resident B cells, but they haven't been studied much in the upper airway. Also, germinal centers, right, where development of cells occurs, are not easy to study because they're in lymph nodes, right? And you can't easily get lymph nodes from people. So here they use nasopharyngeal and nasal swabs. So nasal is when you put the swab just in the nose, right? And then nasopharyngeal, you have to go all the way up more. It's like that's early COVID. They put
I was going to ask. That's early COVID days is my memory.
Yeah. And I'll tell you, I did something. So I looked at the methods for this, and they set it up to 9 centimeters.
Yep.
And that's a ruler. That is how long 9 centimeters is. That's a really long way.
Wow.
The visual that Cindy just gave us was helpful because that is
a ruler. You can put your finger on 9 centimeters on a ruler and then put it to your nose and realize this goes very, very far back.
So I put a graphic in the show notes. Maybe the editor can put it up now because it shows the nose and the nasal cavity. And then the nasopharynx is in the back.
Yep.
Right? Because the front is the nasal cavity and the nasopharynx is all the way in the back. So you have to be long to get there. And also all the way in the back are the adenoids, which are sampled in this study as well, peripheral lymph node tissue, right? So,
and that's something that they removed a lot because I believe I had adenoidectomy when I was younger. That they were just like, oh, you don't need those, and take them out. And then we read this, and I saw all my germinal centers that I could have made in my nasopharynx are gone.
And so, if you remember, as the airway passage goes from the nose to the nasopharynx, it then turns downward, right? And it goes past the tongue, and then separating the nasal cavity from the mouth, there's a palate, which consists of a soft and a hard palate, the hard part being the bone, the soft is tissue. And then as the air goes down, of course, there's a bifurcation. It goes in one way to the lungs, and then there's the esophagus. There's a valve that makes sure when you swallow, it doesn't go into your lungs. And that, by the way, gets weak as you age. And I heard a great talk by an otolaryngologist physician who says, as you age, you aspirate more and more because that valve doesn't close quickly, and that accounts for a lot of infections going on into the lungs. So anyway, I like all this. I think it's wonderful to talk about where things are actually happening instead of saying, "Oh, it's the nasopharynx." That's actually way back. It's not just your nose, right? Although things can happen in the nose as well. Anyway, so they have in this cool study. I don't know how they got people to do this. I don't know either. They got 30 healthy adults to submit to monthly nasopharyngeal swabs for over a year. So the long ones, right? The really long ones all the way in the back. And then they took the samples and analyzed them by flow cytometry, also single cell sequencing. And they could get up to 5 million cells in a single swab. The median is around 100,000 cells, right? And a third of these are immune cells defined by surface antigen CD45. And this includes CD4 positive T cells, CD8 positive T cells, B cells, and then a lot of epithelial cells, which are lacking that immune cell marker CD45. Excuse me. Now, the first thing that is really cool is that these immune cells seem to be stable over time, over the year of the experiment. They have nice graphs showing that. Lots of visuals here. And they see a lot of prominent T-resident memory cell populations by expression of certain surface markers that define those. And they also see B-resident memory cells in the compartment, in the nasopharyngeal compartment as well.
Was the group surprised at the number of cells they collected from a swab? I guess I wasn't expecting to get that many viable cells from a swab.
You would get epithelial cells, right? Because it's scraping. Sure. But I guess
immune cells. Yeah. I guess I always assumed you would need a biopsy, and by non-invasive swabbing, you're not really scraping. So these cells, I guess, either are at the mucosal surface, but maybe they're just more right beneath the epithelium.
Yeah, I sort of couldn't tell how much this was sort of the nasopharyngeal swab versus maybe a less robust swab that might not cause as much cells damage or cells to come
whether
off.
You get that same number of cells if you're just doing the more proximal.
Yeah, exactly. Mm-hmm. At least, again, from my experience with COVID testing, those two feel very different. And so maybe there is a big difference in what you're recovering. And perhaps patients don't often let you do the one that recovers more cells because it's also so much less comfortable.
Yeah.
Yeah, they didn't talk about it.
I was kind of looking for those details more. Yeah. Maybe it's something we could bring an author on. But just where those cells, and I guess, do those cells represent, we think about this a lot with breast milk and the vaginal canal. So you can find cells in breast milk and in the vaginal canal that are in in the mucus. And the question is, are they phenotypically different because they've actually migrated across the epithelium, which is not normally where they are. They want to be like in the tissue. So that's something to think about.
No, it's a good point. If you're just swabbing mucus, right?
Or the surface level of epithelium.
Very surface, right?
Are they at the end of their life and now they're looking to?
Yeah, sure. That's a good point. Yeah. Anyway,
they do later though do a biopsy, so that kind of helps a little bit with comparing.
Validating.
I think that was one of the questions they had, was what you just said, is like, are they really representative? And so going in the actual tissue and taking a sample and then comparing makes a little bit stronger argument, I think.
So they do some single cell RNA sequencing to further characterize these cells. And then they also look at epitopes that are present, because they have a panel of antibodies that have been previously characterized with respect to what epitopes they're binding. And so they take 12 adult donors and do this kind of detailed sequencing. And again, half of the nasopharyngeal cells are B cells, 28% are T cells, 15% are epithelial cells, and then 10% other immune cells. And then they further clustered the B and T cells by gene expression, profiling, enrichment for certain gene sets, and also B cell receptor and T cell receptor sequences. And so they could identify several B cell populations, including memory B cells, antibody secreting cells, B resident memory cells, and others. And they have distinct CD4 and CD8 positive T cell populations, including CD4 positive tissue resident, CD8 positive tissue resident regulatory T cells, Tregs, mucosal associated invariant cell T cells, mite cells, and follicular helper T cells as well. Now, tell me if this is right. The follicular helper cells are usually in the peripheral lymphoid organs, right?
Yep, they're in the secondary lymphoid organs.
Secondary,
important
in the
for
follicle.
helping B cells.
Yeah.
In the
Yep,
follicle,
and the B cell
right.
follicles, right?
And they're somewhere accessible with a swab.
Yeah, right.
So they're out of the
So-
follicle. They're not going to be in a follicle, right?
Or they have
the follicles are,
scraped enough to collect it.
yeah.
Okay.
That'd be interesting to look at some immunohistochemistry to try to see in relation to the distance, where are they? Are they close to where you would, swab?
Sure.
Yeah.
So basically, summarize this part. They see a really diverse set of CD4 positive and CD8 populations in the upper airway.
Yeah, that's cool.
It's very cool. All right. So what about pathogens? So they want to see, this is a portal of entry for viruses. So which pathogen do you think they focused on? SARS-CoV-2.
I'm gonna guess SARS-CoV-2.
Shocking.
Up to this point, I'm thinking, do they know the status of infection of all these people? And of course they do, right?
Yep.
Because I thought in the beginning, are these all naive? In fact, no, there's no way you would be able to get naive infected people who had never had COVID, for example.
Right.
So they did
babies.
babies, right? Single cell TCR receptor sequences on 12 of those NP swab samples. And all of them had been vaccinated, and some of them had what they call a breakthrough infection. And I would just like to say the word breakthrough implies something's going wrong, but it's not. If it's a memory response, it always takes a few days, right? So you're going to get infected. It's not a breakthrough, folks. But they use that phrase throughout.
Yep.
And they can see what epitopes have been represented on their cells by their sequencing and so forth. So they basically, look at T cell receptor sequences and which epitopes are there in the sequences. And
I thought it was really cool that they took peripheral blood T cells.
That's right.
And they did an in vitro stimulation, and they could then sequence the T cell receptors of those that responded in vitro. And then they went and surveyed their single cell data collection for do they have the same TCRs or not, which I thought was a really, really cool way to do that.
That's right. They call that the PBMC-derived personalized SARS-2 specific TCR repertoire data set. Yeah, that's really amazing that they did that. Yeah, that's cool. So all in all, they identified 250 SARS-2 specific CD4 positive and CD8 cells in the NP swabs of these 12 patients. They also found CD8 T resident memory cells that were specific for SARS-CoV-2, and this is in most of the previously infected individuals, and also identified CD4 positive T follicular helper and T resident memory cells in the upper airway tissues as well. A small number of spike CD4 or CD8 T cells were identified in people who only had evidence of COVID vaccination or a subclinical SARS-CoV-2 infection, which is good because they can also see those in infected samples from infected people. So to summarize all this, they find virus-specific memory CD4, TRM, and CD8 TRM cells in the upper airway after an infection, and they demonstrate antigen-specific T cell clones identical in the blood and in the upper airway. Very neat stuff.
Really neat stuff. Yeah.
Yeah,
I was glad
absolutely.
to see some IgA positive memory B cells and plasma cells.
Absolutely.
That's right.
And I think it's important for thinking about, vaccine, because we know that some of these upper respiratory cells are also similar to those in blood. And so it makes us think about, do we need to figure out some vaccine that targets only the resident memory cells, or are they similar to the ones in blood? And so influencing those in blood might also influence those at the local site.
They also extensively look at these germinal center B cells, which they can find, and T follicular helper T cells with both spike and non-spike specificities. And they find the SARS-2 specific T follicular helper cells exclusively in subjects that have had infections. They're breakthrough infections as well. So they conclude, you can reproducibly sample germinal center lymphocytes by nasopharyngeal swabs, which is really good. All right, so now getting to the part that Cindy mentions, adenoids. They're all the way in the back in the nasopharynx, right? They're lymphoid tissues, they're lymphoid organs, and they're unlike lymph nodes because they have a single layer of fenestrated epithelium. It's got big gaps in between. And they have also this fenestrated epithelium has channel-like invaginations that are called crypts. So they said maybe functional germinal centers are present in many adults, and nasopharyngeal sampling may get at some of these lymphocytes from adenoid tissues through the crypts. So they looked at that. They did first nasal endoscopy. So they put a little camera in and put the camera all the way in the back. And you can see the adenoids very clearly, and you can guide, you can take a little biopsy basically of the adenoid to get enough tissue to do it. And so they did that. They sampled in certain individuals, and they could sample reproducibly and stably over time. And they could see, for example, somatic hypermutation going on in BG cells, B germinal center cells over time, right, by sequencing. It's just so cool over the 12 months.
It's awesome.
Yeah. And it was also interesting that is the only site that they saw the germinal center B cells in.
Right.
Because if they tried to sample like lower down in the nose with that same endoscopy method so that they could look in the back and in the front, they only saw the germinal center B cells and follicular helper T cells in the part where the adenoids were, but not in the main tissue. Mm-hmm. So it does seem to support that idea that you were raising, Steph, where, is it just sloughing off in the mucus? It's probably not, because then you would detect them in both sites, but it's probably more so a little bit of damage to it and collecting those cells that are actually present within the tissue of the adenoid.
Yeah. And I guess it speaks to how close the germinal centers are to the surface.
Yeah.
Yeah.
Which is cool. I don't know if I would have guessed that.
Yeah.
And they note that many of the cell types they're finding are not found in blood.
Yeah.
So you cannot use blood to look at those. You're going to have to do sampling of the nasopharynx. And they say, this is interesting, the B memory cells in the turbinate epithelium, it's different, not the adenoid, but the turbinate epithelium, including the B resident memory cells, were more likely to be IgA positive than the same cells in tissue or blood, which makes sense, right?
Yep.
So they conclude immune memory populations are substantially different between blood and two upper respiratory sites, with samples from the upper airway, this is their words, separated by centimeters, demonstrating distinct local immune memory in adults. They're separated by centimeters. So you have to remember that when you do a swab, you're sometimes swabbing really different places, right? Nasopharyngeal swabs. You're never going to get the same exact place. So that can really make a difference in what you find. What about longevity? So they say, how long does a functional adenoid tissue last during adulthood? So they have these subjects, 100 healthy subjects, are aged 18 to 68 years of age. So you can look at the effect of age on it, and in particular adenoid GC activity, germinal center activity. And basically the adenoid B germinal center cells per swab decreased with age, right? With log linear progression. And so as you get old, your B germinal centers back there are becoming fewer and fewer, but you still have some. They say it's not zero. And so this is known that adenoid tissue atrophies with age. And so they say most healthy adults have functional adenoid tissues for germinal center responses. And I'm going to try and wrap this up quickly because it's supposed to be a snippet. But
there's too much good stuff in there.
There's just so much data in this paper. It's amazing.
Adults with and without a history of SARS-CoV-2 infection were enrolled in a longitudinal study with monthly sample collections, all right, to look at what's going on with B-cell memory. And I'm going to summarize this because they were very nice at making a summary statement at the end of each paragraph. Both IgA positive and IgG positive virus-specific B memory cells can be durable in the upper tract for more than 6 months. It's as far as they could go. And these are SARS-CoV-2 spike and nucleocapsid-specific cells.
Following infection, because the vaccination alone didn't do it.
That's right.
So the question is, if you've had COVID now, would you go get any kind of booster within 6 months now that you know this, where you should have reasonable levels of memory B cells that at least some are making IgA in your nasopharyngeal cavity?
Yeah, it's interesting. I think that you get that hybrid immunity, which we've discussed, which we know from literature is superior to just vaccine alone, although we all have that now. You're stimulating two different populations of memory B and T cells because systemic is going to stimulate different sites than mucosal. So, but whether a mucosal infection is going to hinder a vaccine response, I believe the recommendation is you don't get vaccinated, correct?
For 3 months.
For 3. But your question is specific to 6 months. Yeah. I believe the recommendation is that you could.
Yeah, I don't think it's going to mess anything up. I think it's just not necessary.
Right.
Yeah, I think it's hard to know how the systemic and the mucosal compartments are interacting.
Right.
Yeah.
There's one more set of experiments I want to briefly mention. They have identified, of course, B cells, and they've done single cell sequencing that are virus specific. And they note that these spike specific B cell clonal family members all switched to IgA1. So they made the antibodies from some of these. They synthesized, I don't remember how many, but basically from the sequence, they can synthesize and make the antibody. They can put the genes in cells and make the antibodies.
Looks like about 10 or 15 of them.
And they said most antibodies did not bind ancestral spike proteins, the original SARS-CoV-2 virus, but bound Omicron. So these are Omicron primed B cell responses that they're looking at, and they could also see affinity maturation going on in the time that they had done this study. And so they say complex local B cell responses can occur in the upper respiratory tract in response to virus infection, including priming, recall, class switch recombination, and affinity maturation, and development of complex populations of local immune memory, including plasmacytoma B cells and resident memory B cells. So really the most comprehensive, as far as I know, of the this nasopharynx, nasal cavity, and nasopharynx, and giving a lot of information within a 12-month period on what happens. It's quite good.
Do you think that those individuals were not infected with the ancestral strain, or do you think that there's a retention of more recent clones? Because isn't it the case in the peripheral lymph nodes where they've done these studies that they were able to find? I mean, you're kind of the imprinting problem, right? You're boosting, you're back boosting clones from the ancestral strain, but they're not seeing that here.
Yeah, you would think you would have some ancestral clones, but maybe they're so rare.
Yeah,
you don't pick it up with the sampling, right?
And maybe these local populations you have an enrichment in clones that are more recent.
Yeah,
for reasons that would make sense evolutionarily if you need to protect yourself.
Part of what they found was for non-spike antigens, right? So obviously those are not in the vaccine, and those they saw in the nasopharyngeal cavity too. So it could be that the B cells are the ones generated from the infection and not from the vaccine. Whereas if you have the vaccine, it's going to be more draining lymph node and less of those cells would go to this tissue.
Yeah.
I don't think they definitively showed that, but I think their data would support a little bit of that.
Yeah.
And so, yeah, maybe it's Omicron because the majority of them got the Omicron infection.
Yeah.
Right.
Especially
and we wouldn't
recently.
know their history about the ancestral. Maybe they didn't get infected with the ancestral strain.
What I didn't look at was whether they had the bivalent booster, because then they might have vaccine-induced Omicron specific responses.
Well, there was one individual who had this bivalent vaccine.
Yeah, but the rest of them didn't, right?
Yeah, that's right. Yeah.
So if it is Omicron specific, it would have come from infection more likely.
Yeah.
Unless it's cross-reactive.
So let me summarize this. This study shows they find B and T tissue resident memory cells stable over time. The upper airway, B memory and B resident memory cells are mostly IgA positive, with the highest amounts of somatic hypermutation in the B resident memory cells. They demonstrate SARS-CoV-2 specific B memory cells, both in the nasopharynx and the mid-pharynx, the mid-nasal cavity sampling site. These are enriched for IgA positive IgA cells, but there are also IgG positive B memory cells, and they see these for more than 6 months. They get local germinal central responses specific for SARS-CoV-2, and they get evidence for local B cell priming, class switching, affinity maturation, and differentiation in several types, including B memory, B resident memory, and B plasmacytomal cells, they say with signatures of longevity. So this is cool because I always wonder, are there IgA memory cells there? And there are. It's quite clear, which is good. So really nice study.
Yeah, great snippet.
And as they say, you
cool.
can use these methods for other pathogens as well, because they do have a nice established methods in a computational pipeline combining antigen assay with sequencing. It's really cool. And then we could definitely have one or more of the authors back to go into this even more deeply than we have. That could be fun.
That'd be fun.
Yeah, absolutely.
Okay, that's my snippet, which is a little bit of a long snippet, but sorry. Now, Brianne, what do you have for us?
So I have a paper. The paper is called Plasmodium Infection Promotes Genomic Instability and AID-Dependent B-Cell Lymphoma. This is a paper that was published in Cell in 2015. The first author is David Robiani, and the senior author is Michelle Nussenswag. These authors are from the Rockefeller, with one exception of someone who is at the NIH.
Which is Andre.
Yes, which is Andre.
Michelle's brother.
Michelle's brother. Exactly. And I picked this paper partially because I saw sort of a follow-up recently, but realized that we kind of needed to one would need to talk about the basic biology here first. But I also really like that it lets us talk a little bit about a cancer of the immune system. It lets us talk about germinal centers and exactly what's going on in the germinal center, sort of following up on what you just heard from. And it also involves some kind of trade-offs in immunity, which is a topic that I often like to think about, or thinking about, pros and cons of completely getting rid of a pathogen, and also how having different pathogens may impact our immune responses. So this paper is specifically looking at a disease called Burkitt's lymphoma, which is a cancer of germinal center B cells. And typically, when we think about Burkitt's lymphoma, we think of a translocation. So basically, one chromosome being joined partially to another because of some DNA break. And specifically, this is a translocation where a gene called MYC, which is an important gene in allowing for cell proliferation, is fused to some other gene that may turn that MYC gene on all the time. And very often that is the Ig heavy chain. So it's the antibody heavy chain that's going to drive MYC in a B cell. It can also be some other things like Ig lambda or Ig kappa with the light chain. And there are some other mutations that are also involved in Burkitt's lymphoma, like in some cases a p53 mutation, which they talk about in this paper. In the US, there are about 1,200 people per year, and this is a relatively rare adult lymphoma. And in the US, many of the Burkitt's lymphoma cases are either sporadic or immunodeficiency associated. But there is also a third type of Burkitt's lymphoma. And the third type of Burkitt's lymphoma is endemic. It was actually described by Burkitt himself in the 1950s. And it has a 50-fold higher incidence in certain areas of Africa compared to the US, and is 30 to 50% of all childhood cancers there. So in equatorial Africa, this is something like 3 to 6 cases per 100,000 children every year. And generally is peaking in kids who are between 4 and 7 years old. So right away, we kind of see, okay, there's something different going on with endemic Burkitt's lymphoma. And in particular, it has been noted that there is a high incidence of this in areas that are malaria endemic areas. It seems to be associated with chronic infection with malaria. Most of these patients also have an infection with Epstein-Barr virus. And so EBV as well as malaria seem to be part of this. EBV and some of the other mutations are also involved in the other types of Burkitt's lymphoma, as well as being involved with other types of cancers. But malaria is pretty specifically linked to endemic Burkitt's lymphoma. And so in this paper, the authors are trying to figure out how Plasmodium infection may be linked to this B-cell lymphoma in Burkitt's lymphoma. And we understand what's going on with the EBV really well. You can certainly, go to Vincent's virology lectures and learn about herpes viruses and cancers. But why a parasite infection might be linked with a cancer is much less clear.
And the parasite affects red blood cells.
Yes.
By the way, guys, the last TWIP, we did a paper which shows that if you have a worm infection, it severely impairs SARS-CoV-2 immune responses after vaccination.
Wow.
And so there's another effect of a parasite. They're immunosuppressive. But what's cool about that is it impacts the B cell response, but the T cell responses are fine. And so in an animal model, the mice are still protected from severe disease, no matter what virus variant you challenge them with.
Because the T cells are.
But I mean, it does impair antibody response, which is not good.
Right.
But I love that for SARS-CoV-2, the T cells, man, the mice don't die, even if you have a mismatched antibody response. Anyway, so another parasite virus paper. It's very cool.
Yeah, so that's really cool. I'm going to go look at that paper after this.
That's the biggest download of any TWIP episode ever.
Really?
Yeah, I mean, the title is very simple. Worms impair COVID vaccines. And I think that got people in. I've never had 40,000 views on a TWIP YouTube video. It's amazing.
Oh, that's interesting.
Awesome.
Well, and it speaks to what we really, should be focused on from a global health perspective is malaria eradication, malaria efforts to prevent and treat infections. And malaria, I didn't know a lot about it before I joined the Center for Global Health that I'm in now. We have a lot of investigators in the space, and it's a very complicated transmission system. It's spread by the bite of infected Anopheles mosquitoes, which are more active at night, and then the parasites enter the bloodstream, but then they go to the liver, that's where they mate and reproduce before attacking red blood cells. And so there's not a great animal model for the typical malaria that infects humans,
right.
which I think we'll talk about in this paper.
Yeah, they mention, when you think about the epidemiology, this is the Burkitt's lymphoma is usually found in areas where there's endemic Plasmodium falciparum infection in humans. They don't use Plasmodium falciparum in this paper. They use Plasmodium, I don't know how to pronounce
Viroli?
it. Chabudai.
Oh, Chabudai.
Is that because it can infect
Because they can infect mice.
Okay.
Yes.
And that was my question. So two things. Plasmodium falciparum is the most common, but Vivax is increasing. And then, so is what we're going to talk about specific to this, what do you know about kind of how the other parasite strains?
I don't know how Plasmodium chabudai in mice compares to some of the other ones in humans. So I'm not sure I can go from what they've seen here to what you might see in Vivax versus falciparum malaria.
Sure.
But I think that if I think about Plasmodia in general and their life cycle, I could imagine them impacting, splenocytes similarly to what we see in this
cells
paper.
The same, which we're going to, Okay,
right,
cool.
Exactly. But I don't know officially if there are specifics, differences between some of those different Plasmodium species.
And do we know, so when it goes in the liver, Steph, does it infect liver cells or does it have a extracellular life stage?
I do know what are the cells in the liver?
Hepatocytes.
Yeah. So they do infect liver cells. So they do affect hepatocytes. But my memory is that it's like one part of their life cycle, merozoites, right? And then they become dormant in the liver, which are another hypnocytes or something.
So they infect hepatocytes. They become the liver stage, and then they differentiate to merozoites, which are the ones that go to the red blood cells.
Right. The reason why I'm asking is because what we're going to talk about with this paper is a B cell intrinsic activity.
Yes.
But the parasite doesn't infect a B cell?
So far as I know, the parasite is not infecting the B cell. No.
So I think that this is a really cool paper, but it leaves me wondering, how does this actually happen? And you're going to tell us what happens, but the phenotype is a B cell intrinsic activity.
It is.
So it must be stimulation from the malaria antigens, the parasite antigens.
I can hand wave based on one figure, but I probably need to tell you the data in that figure before I hand wave.
Okay, you go.
All right.
So the one other kind of background thing that I want to mention here is that, as I said, this is a cancer of germinal center B cells. So we need to talk a little bit about what goes on in the germinal center, in B cells in the germinal center. Some of this came up during Vincent's paper where he mentioned somatic hypermutation and class switch recombination. So these are things that B cells do after they've seen antigen in the secondary lymphoid organ. They go to the germinal center and they sort of further differentiate to make these high quality antibodies that may have higher affinity and that may have a change in their isotype. So we might switch to IgA or whatever isotype. And a lot of this happens because of the action of a specific enzyme called AID. And this paper is going to talk a bunch about AID, which is specifically active in B cells. And so, as Cindy was kind of mentioning, there's this B cell intrinsic thing going on here. And one part of that is because AID is acting in B cells. AID is a cytidine deaminase. So it acts on cytidines in the B cell's DNA, specifically at the antibody locus, and changes them into uracils. So the cytosines are changed into uracils. Uracil is usually found in RNA, not DNA. And so that is a bit of a problem for the machinery in the cell. And we end up getting some DNA repair processes that have to happen to the B cell's immunoglobulin gene because of these uracils that have been introduced. This can happen in the antigen binding site, which allows for somatic hypermutation and changes in the affinity for antigen. And this also changes the and some switch sites that are in the other sort of part that encodes the constant region, and it will result in class switch recombination. So in talking about Burkitt's lymphoma is a cancer specifically of germinal center B cells. And so there are a couple of things I want to mention about the germinal center and the germinal center reaction that are important. We saw the germinal center in Vincent's paper where he mentioned affinity maturation and class switch recombination. And those are two things that happen to the B cell once they've seen antigen, once that B cell sees an antigen and gone to start making this reaction. And both of those processes happen because of the action of one enzyme, AID. AID is a cytidine deaminase, and AID is able to act on cytidines in specific sites in the B cell. So this enzyme AID is only active in B cells and really only active in B cells when they're in the germinal center. AID is able to act on cytosines and deaminate them to uracils. Uracil is a base that is only found in RNA, not in DNA. So having some uracils suddenly show up in the DNA is a bit of a problem. And so the DNA repair machinery will then act on those same sites, and that's how we get mutations. Those mutations can be in the variable region of the antibody genes, and so we can get changes in the affinity of the antibody for antigen, or they can be in something called the switch sites that are right before the constant region genes, so that we can switch which isotype the antibody is making.
But this depends. The whole story depends on, and we'll hear is mutations are, AID causing double-strand breaks at other places. And I didn't realize that was a thing. Like, I thought that this was pretty specific for the regions you just mentioned, the switch regions and the CDR3s.
Yeah, I did too. I guess when I imagine that AID mutation happening, it's because those sites are accessible chromatin-wise.
Yeah.
At least when I think about the switch sites. So I guess the B cell has some other DNA that's accessible chromatin wise at that same time. And so maybe it's off target. But I also highlighted the sentence where they talked about AID causing off target mutations, because I hadn't thought a whole lot about that before this paper.
Right. To be clear, you don't want to be making random double-strand breaks throughout your genome.
Right. And that's why a lot of the processes in B cells and T cells that influence the DNA have to be so very tightly regulated, because we want to avoid some of these cancers.
Yeah. So the recombination events only occur during that developmental process when they're creating that receptor, and then they're shut off, because messing around with recombining your genome is a very dangerous thing. And that's why it doesn't happen in other cells except B cells and T cells. And then B cells are the only ones that do this, somatic hypermutation, isotype class switch. They're the only cells that can rearrange their genome later.
Yeah, exactly. And
after development.
This may be because I'm teaching VDJ recombination tomorrow. But one other piece of this that I always emphasize with my students is that the cell is very tight in terms of regulating when it turns on these enzymes that can cause mutations and when that cell is going to proliferate. Because you don't want to have an enzyme that's going to cause DNA breaks that is around when the cell is proliferating. You sort of try to separate those two activities as much as possible. And so that's going to relate to my hand waving.
Yeah, that happens with this shuttling between the light zone and the dark zone.
Exactly. Yes.
Right. So, so those processes are separated in time and space.
Yeah, absolutely. So in this paper, these authors did Plasmodium infection of mice. Again, as we mentioned before, it's Plasmodium chabudai. because they were looking at mice. And the first thing they did was just kind of characterize what was going on with the Plasmodium infection. And they saw parasitemia, but they also saw a 63-fold increase in splenic germinal center B cells in the Plasmodium infected mice. And this increase in B cell number was sustained over 10 weeks. They also used an AID reporter mouse so that they could look at different types of B cells. And they saw that AID was expressed in the Plasmodium-induced germinal center B cells. So they have AID positive cells, and they are expressing a lot of, they have a lot of germinal center B cells in this process. And so to go back to the point that you made at the beginning, Cindy, the sort of only hand waving I can make is that somehow this infection is leading to a lot of B cell proliferation in these mice. And so because B cells have this risky mutation process, and we happen to be inducing them to proliferate a lot with this parasite infection, that increases the chances of some problem going wrong in the B cells. But that's the only way I can make this in any way B cell intrinsic.
One thing I thought of is whether the addition of GFP would somehow alter the specificity, but they show here that the AID GFP expression is just in germinal center B cells.
Yes,
so that's
they
good.
do. And they also look at this in, they also just look at AID expression in other B cells as well. And sort of see if AID is really only expressed in germinal center B cells. That's been known for quite a while.
No, the other thing I was thinking about is you did say that it is a chronic infection endogenous. So presumably the children are going to get bit repeatedly and exposed repeatedly and perhaps not fully clearing the malaria at any point. So maybe there's an ongoing overexuberant attempt to create better B cells,
right?
Because it's under this constant pressure.
Yeah.
So it's maybe performing this more than would normally happen for any particular antigen.
Sure. Well, it could be antigen specific in that way, or we could also imagine that maybe there are cytokine changes because of the infection that are just leading to hyperproliferation of all B cells that may or may not be antigen specific. So once they had looked at this, they then wanted to determine if DNA damage was involved with Plasmodium infection. And in some ways, it's a little bit of a needle in a haystack kind of problem, where they, how do you look at all the B cells and find if any of them have DNA damage and sort of find the DNA damage that you specifically look for? Do you sequence the entire genome of every B cell in every infected mouse? It becomes really tricky. So they had to use a system where they had mice with a restriction enzyme site by the MYC gene. And so they could actually induce double-strand breaks at MYC to try to get translocations to happen at MYC. And that meant that they could just sequence the MYC gene and look and see whether it was near other, in the wrong place, whether there had been a translocation. So they had mice with the restriction site at MYC that were bred to mice with a transgenic restriction enzyme that they could control with tamoxifen. So basically, this let them capture anytime MYC got translocated and do sequencing.
And I think one of the interesting things about this method is that I forgot what they call it, like a mega restriction enzyme or something. It's got like an 18 base pair recognition site that doesn't exist in the human genome.
Right.
So it's not going to cut except this very specific site that they introduced into the MYC gene. so that once they introduce it, they create a double strand break, and then they can ask, does it fuse to anything else? Which would be another double strand break, not induced by this enzyme, but presumably induced by AID.
Exactly. And I'm so glad that you explained it that way, because I ended up going on a very long rabbit hole about this restriction enzyme.
Yeah,
and I
went
trying
back
to
and
not
read their prior paper to try to
say
understand
all of that. So you said it perfectly, but avoided my rabbit hole. And they also looked at either mice that had AID deficiency or AID that was overexpressed. They also did a comparison here with B cells that had been activated in vitro from a previous study compared to the B cells that came from malaria infection. And so they did find there were some rearrangements after this. And one of the things to mention is that in the AID deficient cells, they found 191,150 mutations out of 105 million B cells that they looked at. So even stacking the deck with this system, it's a decently rare event. And when AID was present, they had 65,905 out of 105 million. Mostly they were finding rearrangements that were intrachromosomal, so between one chromosome and another. Usually they were recombinations in regions that had genes. They were generally actively transcribed genes. And the number of translocations didn't particularly seem to vary when they, whether or not AID was present. So once you have Plasmodium infecting these cells, you do seem to be getting some translocations.
And I will say, when I got to that point in the paper, I was like, wait a minute, AID is not involved, right? Because it got the same number of translocations with AID. So how do they get the title of the paper?
Right.
Like, that's really weird. But there's a difference in the, I guess, quality and type of translocation that lead more likely to lymphoma in one than in the other.
Yeah, I had the same thought. And in fact, when I was writing notes, I have first doesn't vary with AID, and then I had to go back and be like, this sentence specifically replies to number of translocations and not type. Yeah. Because I was a little sort of confused on that as well. They also, and this is another thing that doesn't really vary with AID, saw that the DNA damage was largely at early replication fragile sites. So they had specific sites in the genome that were being mutated. So all of that was sort of interesting and said that there's something weird going on with malaria infection. But as Cindy mentions, they haven't really brought AID into all of this as yet. And so they looked at sequences of translocations and started to see where do they have translocation, where are the translocations, and does that differ if AID is present or if AID is absent. And when AID is present, they seem to see more translocations of MYC joining to the switch regions, the immunoglobulin switch regions, which are the regions that we know AID influences. So now we're thinking, okay, it's changing the sequence of where we're seeing double strand breaks, though it is not necessarily changing the number of those breaks.
And this is not a surprise, right? So if you have AID, your double strand breaks will likely occur in the immunoglobulin hotspots.
Exactly.
That's not a surprise. So, but I think that what's important is that if MYC goes there, it ends up under the control of an incredibly strong promoter in B cells that drives production of antibodies. And like B cells, the idea is you want to make them an antibody factory. Once you get a good one that you want to make a ton of it. So they become antibody factories so that their promoter of that gene is going to be extremely highly on. And now if MYC isn't there, that's bad.
Right, exactly. This is sort of the worst place in a B cell, perhaps, to put MYC. And we can see that there's a lot of translocation here. So then they further look at, they make some mice with some additional mutations where they either have or do not have AID, but they also have a B cell specific P53 deficiency. And so, you can imagine that this is sort of trying to increase the likelihood of the tumors here and make it so that they can actually sort of see some tumors here. And in the sort of wild type mice, they survive. In the mice who are P53 deficient, whether or not they have AID, you see a loss of the survival of the mice. So the mice are dying. But one of the things that is particularly interesting here is that if we look at the mice that have no AID and no P53, those mice actually seem to be dying because of excess hematopoiesis related to the malaria infection. Those mice aren't dying of cancer. You can sort of imagine those mice with no AID are sort of dying of uncontrolled parasite infection. And so that makes you say, well, AID is probably helping get rid of that parasite. We want to get rid of the parasite. But if we look at the mice who have AID, all of those mice have lymphoma. And so there's this big change in whether AID is present or not in why these P53 deficient mice are dying. Some of the AID deficient P53 double deficient mice do also get some tumors, but they're generally not lymphomas. While all of the AID plus P53 minus mice, and all of these mice are also infected with malaria or with Plasmodium, are in fact seeing lymphomas. And so it looks like this sort of combination of having AID around to cause the translocations that we've seen previously, and a loss of P53 somehow really predisposes you to this particular type of cancer compared to others, and also may have a role in how well you are controlling parasite load.
When I looked at this survival curve at first, again, I was like, what are they doing with this paper? There's no phenotype, right? But whether you had AID or not, if you lack P53 in your B cells and you get infected with Plasmodium, they all die. Right. And I looked at the data first before I read the results section, and I was like, how do they have a paper? But then it became very clear and makes logical sense that if you don't have AID, you can't affinity mature your antibody repertoire, and you're kind of stuck with what you had to begin with, and that's not going to be enough to protect you from the infection.
Right. But on the plus side, you also aren't going to have the risk of mutation potentially leading to these lymphomas.
Yeah, but you're dead either way. So do you die from a tumor, or do you die from the Plasmodium? Which is kind of sad because it really doesn't prolong either way. I mean, they die with exactly the same kinetics.
Right. But it does matter if you are thinking about which thing can be treated.
That is true.
Or what treatment is best.
I mean, you can treat malaria.
Yeah.
Or you can treat the cancer.
Right. So they then look specifically at tumors in either the control mice or in mice that had Plasmodium infection. The control mice had some B-cell lymphomas that were pre-germinal center. They had some T lymphomas, and they had a few post-germinal center B-cell lymphomas that sort of imply that the B-cell got transformed perhaps at the germinal center, which is where AID acts. And when they looked at Plasmodium-infected mice, they saw that most of the tumors were post-germinal center B-cell lymphomas. And so the difference here is that AID seems to be contributing to a particular type of cancer. If you don't have P53, you have some issues, and may end up leading to some kind of tumors, but you won't be predisposed to this germinal center specific kind of tumor. And they looked specifically again at the rearrangements that they saw, and they were able to find some specific rearrangements that are noted in Burkitt's lymphoma, where they can see MYC translocated to heavy chain. They can sometimes see MYC translocated to lambda or kappa light chain, as well as some other types of translocations. And they look specifically at this one tumor, L23, and the specific translocations that are seen in that tumor. And they notice that it really looks like a Burkitt's lymphoma, and has a lot of the features that they see that are sort of characteristic of the human Burkitt's lymphoma. And this is coming from an AID positive mouse. So
in order to get this, they needed to have a mutation in P53, the lack of AID or presence of AID,
right,
and the infection.
Right.
So now if we zoom back out to these children, I wouldn't necessarily think P53 mutations in very young kids. I understand we're moving from mouse to human, but according to this model, they need a second hit of some sort.
Right. Well, one of the parts of this that they don't get at in this paper is that in the endemic Burkitt's lymphoma patients, I am not seeing the number here in my notes, but it's a high percentage also have EBV.
And that so was my takeaway was the hit that you have to have is EBV infection or some destabilizing a virus that causes these destabilizing mutations.
Right. And so, if again, we're getting to an incidence of 3 to 6 cases per 100,000 children. So, the mice, it's all the mice, but they had a P53 mutation. And so you could imagine that at that kind of frequency, EBV or some other hit could be giving you that same hit of P53 loss.
I wonder if there's some sort of connection with nutritional status or anything else that could also make genomic instability higher likelihood.
Yeah, I haven't seen any, epidemiological data on that. But I think that all makes a lot of sense.
I mean, HIV in children.
Yeah, absolutely.
Higher in that region.
I would think that they would note that if there was a correlation with that, they would probably know that.
Yeah, there is already known to be in adults a correlation between HIV infection and Burkitt's. So that's sort of a separate class of Burkitt's that they usually talk about.
But I guess more speaking to just the generalizability of the phenomenon to pediatric populations.
Right. I mean, given the frequency of malaria worldwide, I think it's pretty generalizable just because there's a lot of kids in the world who are at risk of malaria.
Right.
But I think that the idea of sort of this trade-off between controlling the parasite and chronic parasite sort of burden versus potential for lymphoma is interesting. But, before I really read this, I knew there was maybe some hand-waving about Plasmodium and Burkitt's lymphoma, but I didn't have any idea of how exactly a parasite might lead to tumor formation. It's not as clear as what we think about with some of the transforming viruses.
But we still don't know why it's causing genomic instability.
Yeah, we don't really know why it's causing genomic instability. And so we sort of have an idea that, yes, maybe the B cells are hyperproliferative, at least from the beginning. And so my kind of hand-waving idea is, are those B cells just proliferating so quickly in the germinal center that they're not able to turn off AID while they proliferate? It suddenly they're not that the sort of timing split that there normally is. But that I don't have, great evidence on that.
So are there other examples of diseases where you might have chronically active germinal centers?
That is an excellent question. I don't know.
I don't know.
can get somebody to write it.
Yeah. Well, to be honest with you, I'm a little bit surprised that they show in 2015 that Plasmodium gives us these chronically active germinal centers. I would have expected more to be known about that earlier. So perhaps people haven't looked as much as we might have thought.
Yeah.
So, Brian, can you give us a quick summary? Steph, you have to go, right?
I'll stay. I can stay for a couple minutes, but then you guys can wrap up. Yep.
I just want Brianne to give us a summary of this.
Sure. So the summary here is that it has been unclear why there is this relationship between malaria incidence and this particular type of lymphoma, endemic Burkitt's lymphoma. And here the authors show that there seems to be a relationship between Plasmodium infection and this B-cell enzyme, AID, that causes some DNA damage that might predispose cells towards lymphomas if those cells have some other hit, like the P53 hit. And so it's a nice link to how this this parasite can potentially be causing this cancer, and also to thinking about sort of the risks inherent in things like class switch recombination and somatic hypermutation, and these mutagenic enzymes like AID.
So if we could somehow control malaria by vaccination, that would eliminate this.
That would certainly help a lot of these kids. Yeah. Absolutely.
And people are working very hard on that.
Yes.
Yeah.
Okay. It's Immune episode 85. You can find the show notes at microbe.tv/immune. If you have any questions or comments, you can send them to immune@microbe.tv, or you can put them in the comments below here on the YouTube channel. And if you enjoy these programs, please give us some financial support. Microbe.tv/contribute. Cindy Leifer is at Cornell University, at Cindy Leifer on X. Thank you, Cindy.
Thank you. I learned a lot.
Steph Langle's at Case Western Reserve University. Stephanie Langle on X. Thanks, Steph.
Thank you. It's been great.
Brianne Barker's at Drew University. BioProf Barker on Blue Sky. Thanks, Brianne.
Thanks. I learned a lot.
Yeah, I learned a lot too. I always do on all of these programs. I'm Vincent Racaniello. You can find me at microbe.tv. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.